ZombieTech. Welcome to ZombieTech, a forum for engineers, scientists, and inventors to ponder on the technologies needed to survive the inevitable zombie apocalypse.
She's Eddie.
He's Whisker.
And who do we have this week?
This week we have Ellen Jorgensen, and she is a professor at New York Medical College and the CEO and one of the founders of GenSpace, which is this ridiculously freaking cool bio, like, hackerspace / makerspace / community lab in New York.
Isn't that the one that Dory was talking about?
I don't know, but she would know about it.
Because she's one of them bio nerds.
Yes, and we love our bio nerds. Hi, Ellen.
Hi.
So, GenSpace started, was it 3 years ago now? 2 years ago?
2009. We were sort of, milling around in cyberspace on a Google group called DIY Bio. And there had been sort of half-hearted attempts to form a DIY Bio group in New York City. And I typed in, whoever wants to form it, let's meet at the coffee shop near the Beacon Theater on Tuesday at 7 o'clock. And 3 other people showed up, and they became sort of the first core members of GenSpace.
Mm-hmm. And what were the ideas? I mean, so DIY Bio, for those who don't know what that is.
Cook your own zombie. Yeah.
Awkward.
Well, we're not into something quite as large as zombies yet.
Oh.
Darn.
But DIY Bio is a movement. It's like it's part of the maker movement, and it focuses on the discipline of synthetic biology and biotech, where basically reading and writing DNA is becoming much cheaper and much more accessible. The technology is becoming more accessible. And a lot of the people who went through this particular synthetic biology genetic engineering competition known as iGEM, which is held at MIT every year, sort of after they came through the competition as undergrads, they wanted to keep doing that sort of thing as a hobby. And so taking the model of electronic hacker spaces, they started to form these biohacker spaces. And so that's how the whole thing started. So we met and we started doing stuff in people's living rooms. So we would like put plastic all over everything and do something and then throw it all in a bucket of bleach at the end of the evening. And we actually existed for about 6 months in the corner of a hackerspace called New York Resistor in New York.
Sure.
But we really wanted to have a dedicated space because biology, unfortunately, unlike soldering something together, if you're going to actually build a zombie, it takes get myself away. If you're actually going to build an organism, you have to usually do something over a period of days or maybe even weeks. So it's something that you want to have a dedicated space for.
Mm-hmm.
So we set about looking for a dedicated space. And that's really when we formalized and opened a lab.
And so how did you guys, I mean, there's a lot of steps that go between, looking for a space and forming a lab. In terms of equipment, like, where did you guys come up with that and how is it maintained now?
Well, my loss was everybody else's gain because I had been working for a company that downsized my department. And they had a lot of lab equipment that they weren't going to use. And they were thinking of just either putting it in storage or giving it away. And I said, well, I know this great nonprofit. We had very quickly incorporated as a nonprofit in order to receive things like that.
Sure.
And so we got this room full of equipment. And the nice thing about it, of course, is I know where it's been. So I know that there's no strange radioactive residue on it.
Right.
And then we did get a bunch of equipment off of eBay because part of the economic downturn is a lot of biotech companies are jettisoning equipment.
Right.
So that's another aspect of availability that's happening right now for this kind of stuff.
Right. Now, so then is that the same equipment that you guys are currently using?
Yeah.
Okay. And what all does that entail? Like, what all equipment do you guys have?
Well, right now we have 2 or 3 PCR machines, which is a machine that copies DNA.
Mm-hmm.
We have a bunch of centrifuges, water baths. We have a steam sterilizer. We have incubators. We have clean hoods. We have what else? Just anything that you would need to do molecular biology and bacteriology. We have an incubator shaker for bacterial cultures. We have a CO2 incubator for tissue culture, although we don't work with human cells.
Mm-hmm.
We do work with
Zombie
rodent.
cells.
Yes, we do work with rodent cells, and we did do some experiments where we were trying to isolate primary cells from a cow's leg.
Nice.
This was not my project.
Not your leg of choice.
No.
So, with those, like with say the rodent cells and all that, are those lab prepared, or did you guys get a mouse that you found in the subway and
No. No. These were cells that we ordered from. There are organizations that archive different cell lines.
Mm-hmm.
So like Jax or somebody.
It was actually ATCC, American Type Culture Collection. They're the biggest source for any kind of cells in the United States for sure. And they Jax is more for mice, for actual whole animals.
The still alive whole
Yes.
version, yeah.
But the other project was we actually had someone come in who knew tissue engineering, and she was teaching us to isolate chondrocytes from cows' legs from basically slaughterhouses.
Wow.
You have to get them within like a day of the animal being slaughtered, and then you dissect out these cells and then you culture them. And the nice thing about chondrocytes, which is what we were after, is they lay down cartilage. And so Oliver Medvedik, who's the other PhD who's involved in this particular enterprise, was doing this project where you can seed these cells onto a form that's cast in agar, and then they'll lay down cartilage onto the form. And he was trying to make a wristwatch band out of cartilage.
What?
So
that's a little gross, but what?
That's a little zombie-esque. Yeah.
That is a little zombie-esque. I don't know. Do you watch Walking Dead?
No.
No. Okay, that's okay. That's all right.
Sounds like she's pretty busy.
Yeah.
Yeah. I
There's some similar aspects to it, kind of. Yeah. Okay. Well. So then I know you guys have, well, the way your lab is set up, it's considered a biosafety level 1. Would you guys ever consider going up levels?
Okay, wait a minute. What does biosafety level 1 mean?
The biosafety levels are established by organizations like the National Institutes of Health, the Center for Disease Control. They're guidances. They're not laws, they're guidances. They're guidances for how to work with particular types of organisms. And the organisms are categorized as to how much harm they could do to a person. So biosafety level 1 is working with organisms that do not harm healthy humans at all. So that's the definition of a biosafety level 1 organism. Biosafety level 2 are organisms that may affect humans but aren't particularly deadly or infectious.
Mm-hmm.
So the main reason most people have biosafety level 2 facilities is they work with human cells. And once you start working with human cells, you really do have to go to biosafety level 2 because it's just an additional level of control just in case those cells are contaminated with something and you don't know it. So even if you buy them from a commercial supplier, there have been incidents where they have been contaminated with viruses and not
well, like the
deadly
latest one, actually, the latest thing from, was it Michigan or Massachusetts? I don't know. One of the M states, I think, where was it meninges virus? Yeah, that was in oh bollocks, I should have done my research on this. Like where patients got injections spinal injections of this.
Oh, yeah. No, I know what you're talking
Yeah.
about. That's a whole nother ballgame. That's drug manufacture, and that should
Yeah.
absolutely never happen.
Right.
This is materials that are sold for research, and so the research grade materials are checked less stringently than something that's for use in humans normally.
Mm-hmm.
And so once in a while, they've had instances where the cultures have actually had some sort of contamination in them. So most universities, for example, even if the cells themselves are considered biosafety level 1, they're absolutely bound to treat them as biosafety level 2. And certainly any blood products, things like that, are all biosafety level 2. Any tissue samples, all of that. So it would be opening the door to a whole nother line of things that we could work with. And I've certainly thought about it, because it would be nice to have the capability of doing more biomedically related research, but then it also opens the door to people saying, well, couldn't somebody develop couldn't they work
deadly
with
virus.
viruses?
Yeah.
Yeah,
Blah blah. Yeah.
People have such I think erroneous assumptions about how easy it is to develop something. It's not easy to make a human pathogen. It
I
really
take
is.
a very simple approach to people like that, because we have to talk about topics like this all the time, doing what we do here on the show. And if somebody's a little bit nervous about biotech, I just simply explain to them that literally 1/12 of their own genome is leftover viral material that could potentially wake up if it bumps against the wrong thing.
That's nice.
Way
Do you
to
also
where to
tell
scare them.
So, they're a walking
Sack
little
of
time bomb in their own mind now, going through the world, and they're like, okay, I myself am
way to
just
induce OCD there, Whisker.
Yeah, well, it's true. You want to be nervous about what can happen in a little lab like that. What can happen in nature is far more scary to me.
And you also know that you have more bacterial cells on your body than you do human cells.
Yep.
Tasty.
Yep.
And the scary thing is when a person dies or becomes immune compromised, it's amazing how fast those creatures can take over you.
I mean, that's why the zombies turn almost instantaneously, right?
Yeah.
It's kind of
That's
crazy.
why they're greenish,
within 2 seconds. Yeah. All of a sudden they just look really ugly, and flesh is suddenly just decomposing.
Yeah, bacteria not
faster
good
than
for your
corpses.
complexion.
No. I guess not. So it was interesting because I mean it's fantastic that you guys are have this professional grade equipment. And we talk to a lot of open source hardware folks, a lot of tinkers, hackers. Is DIY lab equipment something that you guys would be able to or be willing to develop / incorporate in your own lab?
Well, this community has produced a few different pieces of reverse engineered equipment. Probably the most famous is the Open PCR machine. We have one of them here. It looks extremely DIY. The outside is all laser cut wood, and it looks very homemade. You get it and it comes in a kit and you put it together. The thing about GenSpace is that the people that founded GenSpace, the technical people, were molecular biologists. I think a lot of other DIY groups that have popped up, the main people have been more engineers. And so they tended more towards what they could do, which was reverse engineering the lab equipment.
Mm-hmm.
So you have companies like Pearl Biotech coming out of BioCurious on the West Coast, making the Open PCR, they made a gel box first. And that's all well and good. Because that isn't our area of expertise, we haven't really segued into that. We did do a DIY microscope based on some plans that we had. I think it was Hacteria had done. They took uh, one of those little cameras that a webcam,
Yeah.
and if you turn the lens around on it, it's a really simple hack. Then it becomes a magnifier, and you can actually make a little microscope. It's just you have to figure out how to put it on a platform so you can focus it.
Right.
And that's sort of a typical reverse engineered DIY hack. But we'd like to get into that a little more, but I think we just need to get some members who are more interested in that.
Sure. So you guys wouldn't be like, you guys would entertain like using that type of equipment in the
yeah,
lab?
I mean,
Okay.
we use the open PCR, and we've done some. It's where it's really good is when we go out and do workshops on the road, because it's a lot lighter than a conventional machine.
Sure.
And
Yeah.
if we lose it, it's only $600, not $6,000.
Right.
And the other thing is the Dremelfuge, which is Cathal Garvey's invention, where you use a CNC printer to print this little rotor out of plastic, and you fit it into a Dremel tool, and it becomes a microfuge.
Oh my gosh, there are so many ideas going through my head right now.
that works very well.
Because we're actually trying to set up a hacker space in our basement, and we have a lot of the electronics, the woodworking tools, crafts and such, but I'm a bit of a bio geek myself, and oh man, having my own lab would just be maniacally awesome.
You should check out Cahall's website, because his spare bedroom is a lab. He actually, since he's in the United Kingdom, he's in he's in Ireland, and you need a license in that part of the world to practice biotechnology, to do genetic engineering. And so he actually got a license from the government, petitioned and got a license. And if you see the photographs of his spare bedroom, his sterilization equipment is a pressure cooker. His incubators are styrofoam boxes with electronics, probably Arduino controlled or something.
Sure.
Heaters. He's got Dremelfuges as centrifuges, and the open PCR. So he's completely all of his equipment is DIY.
That's very cool. Very, very cool. I'd be a little scared of things leaking, but
well, the Dremelfuge, it's funny. Whenever we've used it, we've sort of taped it to the side of a table and kind of stood on the other side. but yeah, it really isn't that hard to pick up used equipment on eBay. And my dream eventually, I would like to see this be kind of like the Fab Lab thing, where you've got certain key pieces of equipment that you need. I wouldn't say that the genetic engineering is for everyone, but the DNA analysis is something that you can do on your kitchen table, and it's perfectly safe. And let me give you a couple of examples, if you don't mind, of this, because people say, well, why would I want to set up a personal lab? Well, one of the things that I did this summer was I identified an invasive species in my backyard. There are these things that look like ladybugs that sort of infest the house during the winter. They all come inside and they live up in the corners of the windows. And because they looked like ladybugs, I didn't want to kill them. I was like, well, these are good for the garden. And then I heard a rumor that they weren't ladybugs, that they were an invasive species from Japan. Now, the only way for me to test this that I can think of easily, outside of sending them to an entomologist, was this technique called DNA barcoding. And all of the stuff I'm going to talk to you about is done using a PCR machine. So you extract the DNA from the bug, and you do a PCR reaction, and then you mail it out, and it gets sequenced, and you get the sequence back. And you can plug that into the public databases. There are huge public databases. The National Center for Biotechnology Information, which is your tax dollars at work, has enormous databases. And it came back with the answer that, yes, this was this Japanese beetle and not a ladybug. So I found an answer to a question that I wanted the answer to. So that's one. There was also a very highly publicized study that 2 high school kids did a few years ago. The press dubbed it Sushi Gate. They went and they took samples of sushi from restaurants in their neighborhood, and they sent it out. They did PCR on it. They sent it out for barcoding, and they found that something like 25% of the time, the fish wasn't what it was supposed to be. So they were serving things like calling it white tuna and charging outrageous prices, and it was tilapia, which is a pretty cheap fish.
What?
Yeah.
Oh my
And then
gosh.
so you can do a lot with
Wow.
truth in food labeling
Yeah.
using that. You can also check to see if you're radically opposed to genetically modified food. You can check. So there are certain markers in certain types of GMO food, like the Roundup Ready gene, that you can test using PCR. So you can test your food for that. The other thing, I love this story. There was a journalist in Germany who, Sasha Carberg, he decided he was going to make a movie about biohacking. And as part of the movie, he and a friend tricked out a van as a molecular biology lab, and they did this DIY forensic study. In Germany, people are they're very concerned with the streets being nice and clean and everything being orderly. And in his neighborhood, somebody was letting their dog poop on the street and not cleaning it up. So they decided to do something about this. So they took a sample.
I could see where this is going.
Exactly. They took a sample of the poop. They extracted the DNA. Then they went through the neighborhood with tennis balls and they threw them to all the dogs, the local dogs. They extracted DNA from the saliva on the tennis balls. They took photographs of the dogs. They have this great picture in the movie of them hanging out of the van with telephoto lenses, snapping pictures of these dogs. And then they did a DNA fingerprinting exercise, just the way they do in forensics cases with humans. And they were able to identify which dog had crapped on the street, confronted the owner, and made her clean it up.
Can you imagine somebody knocking on your door?
Excuse me.
Your dog pooped on the street, and no one else lets their dog. Well, how do you know it's my dog? Well, see, we tested its DNA. We have photographic evidence. We have the tennis ball, Exhibit A. Yeah, that would. Yeah. Kind of feel bad for the person whose dog it was, because that is a difficult doorbell to have to answer.
That's great. So, I mean, all these things, like for a lay person, someone who's,
now as a lay
never...
person,
yes,
I can say that
Oscar.
The ladybug thing,
yeah,
you can solve that by having the gene sequence to actually test and make sure. But as just sort of a lay person, the ladybugs are really red colored, and the Japanese beetles are more of a dusty, dirty brown orange color. They don't have as high of a contrast, so it's pretty easy to tell the difference.
Whoops.
Yeah, so I don't know. I just look at them and go, okay, that's a ladybug or that's not a ladybug. But we've had a problem with them for a long time in the Midwest, so the eye is trained to be able to tell the difference.
See, my eye's not trained, so
yeah. When they swarm, it is epic. They are all over everything.
Yeah.
Anyway, I don't want to get too distracted with them.
So but they have actually taken butterflies that they've thought were one species, and through this sort of genetic analysis, they've realized they actually were several different species that looked very much alike. So much alike that taxonomists couldn't identify the difference until they did the DNA. So it is useful sometimes.
Yeah, yeah. So for the layperson, like a lot of what you're talking about sounds kind of intimidating. PCR machines, DNA fingerprinting, forensics, etc. So like in terms of the folks who come to GenSpace, are they mostly like bio nerds, or are they kids, or like noob adult learners? What kind of demographic do you end up getting?
We actually, for the first 6 months, we surveyed everybody and we asked them, what they did for a living and why they were taking the classes and everything. And we teach these introductory biotech classes, introduction to synthetic biology classes. And it was really interesting. About a third of the people are artists, because in New York, obviously New York is a big art scene, and there's a lot of interest in the arts community incorporating some of this technology into their artwork. So about a third of them were artists or designers or architects. About a third of them were engineers or computer science people, because the idea of coding life is something that's very attractive to those folks, or engineering life.
Mm-hmm.
So it's kind of the next step, the next frontier. And then a third were all different professions. We had people who were winemakers or worked with fermentation and wanted to explore the possibility of improving the organisms that were doing it for them. There were people that were kind of support for scientists, like we had a paralegal who worked in a patent law firm. We had bioinformatics people that had to interact with these scientists and wanted to know a little bit more about what they were talking about, sort of from a hands-on perspective, because all our courses are very intensely hands-on.
Mm-hmm.
We had venture capitalists who said, I want to know what they're doing with my money in those labs, you
Sure.
know?
Sure.
So it really was quite an interesting, and we've had everybody from about age 13 up to retirees.
Nice.
So I'd say the average age is somewhere in the early to mid 30s.
Okay.
And and we do get a lot of students. We give them a discount. So either college or graduate school.
Mm-hmm.
We don't tend to do a lot of teaching to people under high school age, just because it's a little bit sophisticated and it's not really part of the curriculum.
Mm-hmm.
So
and it's pretty expensive technology that you guys are working with, so.
Yeah, and the attention span of a middle schooler is just not conducive to doing this sort of stuff.
Mm-hmm.
I mean, occasionally we'll get somebody who's really outstanding. Sometimes we get kids and parents taking classes together,
Mm-hmm.
and that's fine. But we're not really focused on anything below high school level.
Sure. Now, say somebody who's completely new to biology, well, besides their typical high school lessons that they took like 10 years ago or what have you, or 20 years ago, they take these courses. Are they then signed off on techniques so that they can work autonomously, or how does that all work?
Well, we sort of decided that when we incorporated as a nonprofit, we had to develop a mission statement. And I feel very strongly that this technology is so important that everyone should have sort of a basic working knowledge of it, even the way they do of something like electricity,
Mm-hmm.
because it is really going to pervade just about every aspect of our lives in the very near future. So, and there's so many sort of ethical decisions that have to be made about this technology. I really feel more comfortable knowing that people aren't demonizing it. It's not mysterious. It's something that they've done and accept. So I like to see people do a lot of hands-on stuff. So the people that come through here normally are starting at pretty much ground zero. We do get the occasional biology student who just wants more hands-on stuff,
Mm-hmm.
or someone who worked in the field and then got out of it and wants to brush up on their skills. But I'd say the majority of the people start out with pretty much zero. So we start from what does DNA look like, and then we go on from there.
Mm-hmm.
And when they go through the class, they do the techniques hands-on, but of course, in one class, it's not really something that they could probably do on their own.
Mm-hmm.
So sort of the two parts of our mission are, number one, to do outreach, which we consider the class as outreach. But the second part is to provide a space for people to do independent research and to innovate at a low cost, and to provide mentorship. So we have a membership just the way a hackerspace does. So for $100 a month, you can come to GenSpace and you get the use of the facilities, you get safety trained, you get trained on the equipment, and you get to bounce ideas off of us, the people who are the PhDs who have a certain amount of knowledge in the area. And what a lot of people do is they either have some idea that is kind of half-baked and we throw it back and forth for a while,
Mm-hmm.
and eventually it becomes something that they actually want to do. Or a lot of times we have sort of group projects so they can start working. Right now we have a project where we're trying to make some standard DNA parts for a course that we're teaching to build a biosensor. So there are 3 members that are involved in that, and they're learning while they're doing it. So that's kind of how it goes, is usually people come in and they help someone else, or they do a project, but we help them with it until they can become independent.
So, you brought up the wine guys, beer guys, stuff like that, and that's very fascinating to me. Not the winemaking or anything like that, but the important thing to note there, I think, is that a lot of the people who are approaching this are really doing it for all the same reason. There's this watershed moment in biotech that has happened. We can now know for certain the truth about something, and that's new for biotech. Fairly new, within the last couple decades here. Before, it was a lot of compare and contrast to figure out what was what, but now they can actually sequence it and know for sure, and that's really, really interesting. And that's sort of like for all the electrical engineers that listen to this show, imagine trying to work without a voltmeter. That's what they were stuck doing, but now they have it and they're empowered, and I think that's great.
It is true. It's amazing that the so-called deep sequencing, which is sort of the next generation of DNA sequencing, has changed everything, because and this is much more recent than 20 years ago. This is in the past, I don't know, maybe 5 years, that you can fairly easily get the sequence of everything in a sample, everything, even the most minute quantities of DNA. And for the first time, for example, the human microbiome, all of these bugs that are living on you and in you, we never could really characterize it before, because if you tried to just take a sample like a swab from your mouth and grow it out on a plate, most of the organisms that live in you can't be cultured on a petri dish. So there would be things there, but you wouldn't see them. You would just see the things that you could grow. And so you were biasing what you were seeing. But now you can have a completely unbiased view of everything that's in you. And it's really opening up a whole other science of health, because what organisms are living in you has a lot to do with your health and different disease states and all sorts of things. It's really fascinating.
Yeah, and I'm a nurse as my day job, and I know we deal with C. diff.
Yes.
Yep, a lot, and that's mainly just because of the imbalance between gut flora, really, that happens as a result of antibiotic
gut flora
administration.
Yeah.
So, I mean, yeah, as soon as something's out of whack, you're diarrheaing up.
I think a lot of lay people don't realize just how important gut flora is. You need to have that bacteria in you. You need to have all those little guys down there doing their thing.
Yep.
Because your body really doesn't know how to do all that much with what you're processing, and they help a lot.
Yep. So, in these classes, sorry, just to backtrack a little bit, in these classes, what sorts of techniques do you guys teach?
Well, my class, the sort of the biotech class, what I try to impart is the 3 basic things that enabled the genetic engineering revolution, which the whole field of biotech. The first was the ability to transfer DNA from one organism into another, and that's called transformation. And so we do kind of the standard biohack, which is we take a teaching strain of E. coli bacteria, which, by the way, is not closely related to the E. coli that is in outbreaks. It's sort of the difference between a Chihuahua and a pit bull. So we take that and we put a gene that codes for a green fluorescent protein from a jellyfish into it. So we turn them fluorescent green.
So cool.
So that's the first technique is transformation. And the second one is being able to cut DNA and then paste it together. And the thing that enabled that are these really cool enzymes called restriction endonucleases. They originated as the immune system of bacteria. Since bacteria, they're just single cells, they don't have the same type of immune system that we do. But what they've developed are these enzymes that cut invading DNA. And the way that they do this is they scan the DNA for a particular series of letters, and the bacteria hides that series of letters in its own genome by attaching what's called a methyl group, a carbon and hydrogens, to the DNA around that sequence. And so it acts kind of as a smokescreen, so the enzyme doesn't recognize it. And so if it's invaded by a bacterial virus, these enzymes will cut the viral DNA into little pieces and inactivate it, but its own genome will be safe. And once people realized that these enzymes were like molecular scissors and that they had a specificity, they used those. They used those to make constructions. Basically, that's another really huge technique, because before that, you couldn't really, you're working with something that's infinitely small. I mean, to you, you can't see it even under a microscope, and you can't really manipulate it. But now you have this enzyme that cuts exactly where you know exactly where it's going to cut. And that was really important to be able to piece things together, because if you're going to make something, you want to edit it like a film. You want to edit it together. And you can't do that without these enzymes. So that was another huge discovery. So we do that. We play with DNA, we cut it up, and we splice it together. And then the last technique is the PCR, which is a way of kind of Xeroxing the piece of DNA in a tube that you're interested in. So your entire genome is 3 billion bits of information. And that is a huge amount of stuff to deal with. A lot of times, all you want to look at is a very small portion of the genome. For instance, if you want to know something about your ancestry, you might only look at some genes. If you were a male, on the Y chromosome, and you would want to kind of home in on that particular place. And PCR is a way of amplifying only the part of the DNA that you're interested in, way above the quantity of all the other stuff in the test tube. So it kind of drowns it out. And it's like the third major technique that we teach. Also, something called gel electrophoresis, which is a way of analyzing DNA, separating it by size. So those are sort of the things that they're standard molecular biology and cloning technologies. And and along the way, we extract DNA from bacteria. We grow bacteria. We sort of work with it. And then we also teach the synthetic biology course, where you actually use these enzymes and these pieces of DNA, and you build a biosensor. So
biosensor, what, like
something like if you shine blue light on it, it does something like turn purple or.
Whoa.
Yeah, that's what Scott does at work, I think. He uses all the different lasers and all that to fluoresce different.
Yes, yeah. That is true. That's true. So let's think about the zombie apocalypse for a little bit here, because
previous portion of this show was brought to you by really big words that are really cool.
So we're making, like, you guys get to make these fluorescent bacteria, E. coli, right?
Well, forget that. She's got enzymes that can.
I'm thinking of
cut
lighting
up
mechanisms.
DNA that matches certain patterns. That sounds like an effective zombie attacking weapon right there.
Yes.
I would like to put forward that the skills that we teach are actually probably some of the most critical in averting a zombie apocalypse, but and maybe even ameliorating it after it happens. I mean, using big words too, but definitely. I mean, hey, just come on. Just about every science fiction movie. Hey, War of the Worlds. Hello, it was a virus.
Yeah,
we can engineer those. We've got an app for that.
We have an app for that. So okay, let's posit this. Let's say the CDC implodes, okay, and who knows why? Nobody knows why. And we've got these labs all over the place, and then we have GenSpace. Now, obviously, in our current day and age, being that you guys are biosafety level 1, you probably can't handle the zombie virus. But let's say the whole world has gone to hell, right? We're dealing with zombies. What could GenSpace do, given your expertise and your equipment?
Well, certainly, if everything implodes and there's the zombie apocalypse, I don't think we're gonna be quite so concerned about what biosafety level we're working at. And I would really like to think that some of these DIY labs could do some pretty quick turnarounds of a virus that might be able to target zombie physiology. Now, we'd have to study a little bit more, but I'm sure that since they generally shed pieces of themselves as they ambulate, that there would be plenty of tissue samples to work with in this case. So I would try to make some sort of a virus that would target zombie-specific proteins. So we would definitely have to use a viral delivery system, I think. But
excellent.
We definitely would be able to do something.
Like, say somebody dropped a sequencer off at GenSpace just because it's the zombie apocalypse. I mean, I imagine with something like that, you would be able to, instead of having to send that sample out, you could take care of it at the lab and then create some sort of anti-zombie virus. And
well, there are sort of manual ways to do sequencing. When I first started, it wasn't done by machines. It was done by electrophoresis, and it was done pretty much manually. Mm-hmm. So it can be done. It just is tedious. But, we might be reduced to that. We do have a synthesizer here. It's old, but we could get it back up and running. I think it's a synthesizer. I can't remember now if it's a synthesizer or a sequencer, but definitely we would be able to get something like that up and running.
Dude.
I mean, hey.
You guys could be the cure to the zombie apocalypse virus.
Now, in all the hypothetical versions of this and stories and movies and stuff, it seems like the cure is, either the systematic shotgun to the head cure, but the more biological approach is to sort of create a counter virus that knocks out whatever's causing the zombie one and either drops them or cures them. But I'm really interested in this whole enzyme thing, because that seems like it might be a much more easy to weaponize approach. What would you need to do to be able to design one of these enzymes to attack a particular gene sequence? How trainable are they? You said they're sort of a tool that's kind of general purpose at this point?
Yeah, the thing is the sequences they recognize are mostly pretty short, and so they occur fairly frequently over a large genome like a human genome. So unless you could somehow engineer them, and at this point we're not quite there yet with protein engineering, but at some point in time we probably will be able to engineer them to be a little bit more actually, you're giving me an idea. I wonder if they could be trained to somehow work in concert so that you would have to have a number of different enzymes or a number of different sequences
sort of like
all together.
Key that has different
tumblers. Yeah,
exactly.
I don't know. That might be very interesting. You still would have to get them into the zombie cells, which means that you probably are stuck with infecting them with a virus that has the code for the enzymes in it.
Mm-hmm,
so again, you're stuck with some sort of viral delivery. but that, you could certainly use the backbone of a virus that already infects semi-humans. So
let's get on that DIY biotech, people. We wanna make these enzymes a little bit more of a general purpose tool, a little bit more programmable.
What CNC?
If you want to look at it that way, I look at
Viruses.
biology as just molecular, chemistry. So it works the same way as my programming code does when I'm doing that sort of thing. If you want to approach it that way, it's just a lot more analog, which is great.
Yep.
actually, you've been anticipated because I believe, if I'm not mistaken, the original Blade Runner movie, when they were running through all the things that they could do to get rid of the replicants, I think they did mention restriction enzymes.
Nice. Good minds think alike. So actually, my original idea was just that you could, because we're setting up a zombie apocalypse bunker, so we need lighting, right? And if we don't have, what's it called? If we don't have all the power in the world, we could theoretically use glowing E. coli, don't you think? To light up the corridors or something?
Sounds like a really bad idea.
Well, as long as it doesn't leak or crack.
Actually, there was a team of students in that iGEM competition that from Cambridge that did a project where they engineered the proteins that create light in things like fireflies,
Ooh.
and they made, not only did they make it much brighter than it normally is, but they managed to change the coloration of it so that they not only had lights, but they had different colored lights. The only thing about those systems, though, is they're using a lot of energy to create that light. So you have to keep feeding the bacteria, and you also have to aerate them. So you end up with something like almost like a lava lamp, where you're bubbling stuff through it in order for it to produce the light.
Could you like feed it zombie flesh?
Now
That'd be super gross, though.
that would be super effective, I think.
There is a high energy concentration in, animal cells and whatnot. So I don't know, but do you really want to train a little critter like that to eat human flesh? I mean, that's just...
eating bacteria. I think we already have that.
Flesh-eating glow sticks. It's a whole new way to rave, baby. Oh my gosh! All right, I just have this gross picture in my mind of like a vat of glowing stuff, and chunks of zombie flesh down there. All right.
We've had some terrible ideas on this show, but I think that's right up there at the top of the list.
Soylent Green.
Well, it is. Kind of. Yeah. So I wanted to touch on this before we asked our two super questions. So we talked to a lot of open source hardware and software folks about patents. And what is not always known is that there are also pharmaceutical companies and biotech companies who are looking to patent genes. And I know that you had an opportunity to talk to some folks about the company Myriad, and their desire to, I think, patent the BRCA genes, right, for breast cancer.
Okay, so what happened was Myriad Genetics developed this test for the BRCA genes, which are genes that if you have certain mutations in them, a woman has something like a 10-fold elevated risk of breast cancer, and it's passed down through families. So it's an important test to get, especially if you have a history of breast cancer in your family, because you can do some proactive things. I mean, they're pretty radical, but people do them, like get bilateral mastectomies and things like that.
Mm-hmm.
But it's a pretty serious risk factor. So this particular company has done some things that aren't sitting very well with breast cancer advocacy groups, because for instance, they've only licensed one laboratory in the United States to do the test, which means if your insurance company doesn't subscribe to that lab, then you have to pay something like $3,500 out of pocket for this test.
Jeez.
So the hearing that I went to was at the U.S. Patent and Trade Office, and they open certain things for public comment periodically. And so it was an open hearing. Anyone could come and just sort of speak their mind. What happens in reality is mostly the people that come are corporate dweebs and people that are very deeply involved in whatever this particular struggle is. So you had people from the biotech industry, you had lawyers, you had someone from the American Association of Pathologists that was actually bringing the suit. You had the breast cancer advocacy groups who were against Myriad sitting on this technology.
Mm-hmm.
And then you had me, which was I kind of came out of left field. And basically, I sort of took it up a notch and said, well, there are those of us who are exploring our genomes ourselves, and we're uncomfortable with the fact that Myriad's patent, it prevents me from exploring my own genome.
Mm-hmm.
We're not talking about someone who's doing a test to make money and selling it to someone else. We're talking about someone who wants to just look and see what's going on in their own body. And we're being prevented from doing this in a DIY way, technically by this patent. Not that they actually would ever go after us, and it just seems wrong. And one of our advisory board members who's at the Genomics Center at Cornell University, his name is Dr. Chris Mason, he did a study and he kind of looked through all the patent literature. And do you know that virtually 100% of your genome, the parts that code for proteins, is patented, is covered by some patent? 100% of the genes in your body are covered by patent at this point. So it's really gotten sort of out of hand. And it's gone, this Myriad case went all the way up to the Supreme Court, who threw it back down to the more local court and said, we really suggest that you overturn the right to patent nucleic acid sequences, because it's like patenting a hand or a foot. It's not something that should be patented.
Right.
But the lower courts have been traditionally much more industry friendly. And of course, if you're a biotech company or a startup and you want venture capital, to say that you have something patented is a way to raise money.
Mm-hmm.
So some of the testimony was from people who said, look, you would gut the entire biotech industry. You would topple it if you overturned stuff like this. So
sounds like a good plan to me. The reason why I say that is because throughout human history, almost every major revolution in countries has been because of food, cheap food. You take away cheap food and people revolt, okay? In this country, we have McDonald's, okay? So that's not a problem. But if you take away people's ability to cure their mom or at least know their mom's going to get breast cancer, and all of the other things that this biotech sequencing and all of that opens up for people, you take away the future's version of cheap food, the ability to fix yourself, to go down to the human body automotive shop, right? And you put it in the hands of a very small group of people. And when the control of food sources has been in the hands of a small group of people, it has always caused civil unrest. That's not going to be a problem here anymore, but this new important technology, the ability to fix yourself when you break, I think is just as important as food or water or air. The access to those kinds of things, this is as important as those things, and I think it's enough to incite people to be very misbehaved. We may not be talking about revolution here, but people will get this stuff one way or another, and you want to have it be done in a way that's sensible and is in the hands of good doctors and PhDs who know what they're doing, rather than back alley cyberpunky mods and whatnot. You don't want to encourage that kind of a black market. So yeah, patenting, especially these things that involve the human genome stuff, is a really, really, really bad idea in the long term, I think. Just looking at human history, I think it's a bad idea.
The other thing
what happened in
is
Prohibition or whatnot, you make it illegal, it goes underground, people make a lot of money off of it, giving it to the people who are going to get it one way or another. So,
Mm-hmm. And the other thing is a lot of this research, very little of it was actually done by the companies. They usually buy it from a university.
Mm-hmm.
So it was the original research for these uses for these genes is all done with public money, because most of the research in the United States is done with the biomedical research is done with funding from the National Institutes of Health or American Cancer Society. It's all money that the public has donated for research or that comes from tax dollars. So to have this stuff then make some company rich is also something that really bothers me.
Sure.
do you think that showing up and voicing the voice of reason had a positive effect there?
I think that it's interesting that the patent office and the people that work there are actually very grateful when the public pays any attention to what they're doing. And I think they liked the fact that somebody from the public cared enough to come and testify, because really what they see over and over again is kind of the same old usual suspects saying the same old things. And it's really boring for them. That said, I don't think that I changed anything all that much. I don't know. It'd be very interesting to see how this whole thing eventually plays out.
Yeah, you're up against a $500 billion lobby, so good luck with
Exactly.
that. Okay,
So-
are we going to get to our
Yes. And
top secret questions?
there's
Well,
at
at least top secret to the guest. All the listeners know them already.
Right. So, 2 questions. Here's the actual ZombieTech interview. First question. What 3 tools would you bring to a zombie apocalypse bunker?
These should be good, considering that her idea of tools is a little bit different than most people's.
Correct.
Well, I would say I would like to bring a DNA synthesizer.
Fantastic.
I definitely would like a DNA synthesizer. And I think I would like to bring a CO2 incubator.
Okay.
I want to be able to grow those cells and do those extractions. 3 tools. This is tough because in biotech you use a lot of different tools.
Yeah.
But
It's
there's
important to bring friends and let them bring 3 other tools.
Yes.
Find, yeah.
Yes. But it's also good because there's some tools that you can easily make, like the DIY centrifuges, and other tools that are going to be harder to make.
Mouth pipetting.
Ooh. Yeah.
There is
maybe-
no
maybe
mouth
that's
pipetting in
allergy
the
kit.
zombie apocalypse. That's really
Maybe
bad.
a set of pipette men. I don't know.
I love how on those OSHA things that everybody has to do, they're like, no mouth pipetting is allowed. I'm like, when's the last time I decided to mouth pipette bacteria? About never. So we have DNA synthesizer, CO2 incubator, and was it pipette man?
Yeah, this is really tough.
I don't know.
though.
I think
It's okay, I mean, it doesn't-
glassware
it's
sort
up
of
to you.
stuff would be like
It's totally up to you.
let's go borrow that from the local lab sort of thing. I mean, I wouldn't use that as my tool.
Yeah, I mean, yeah, so we'll go on scavenging parties,
Well, it would be nice, certainly, to be able to sequence it once you'd made it and just make sure. But I think a lot of other stuff could be DIY'd. So, and something like PCR, the copying thing, they used to do that just by repetitive dipping of tubes into water baths at various temperatures, because really it's only a thermal cycler. So very tedious, but you could, boy, if the zombies were beating on the door, I would spend an hour dipping tubes in a water bath.
Yeah, got better things to do.
Yeah.
Yeah. All right. So those are the 3. And
So is the water bath the last one or no? Wait.
No. I think that probably a sequencer would be the last one.
Okay.
You synthesize it and you sequence it to make sure you have what you think you do.
Got it. Sequencer, synthesizer, CO2 incubator. And you pretty much guessed the second question, which is, what book would you bring to the zombie apocalypse bunker?
Yes.
What book would I bring? I have to just bring one. This is terrible. Well, obviously, if I'm going to be doing anything about the zombie apocalypse, it would have to be Sandbrook. Sandbrook is the molecular cloning manual. It is the
awesome.
Bible for molecular biologists. So it would be Sandbrook.
Awesome.
We've had some pretty good ones over the course of the show here, and that is an excellent addition to the library, I think.
All the electronics guys are gonna be like out of their league for once. So
well, it was either that or like one of the classics of Zen. If I'm just gonna sit and be eaten by a zombie.
Yeah, you can tell a lot about a person's like emotional state, demeanor, whatever, by if they choose something to rebuild society or they choose something to entertain themselves while it all burns.
Right.
So, are there any sites that we can find more information? Well, obviously there is, but what sites can we find more information about GenSpace and about your own projects and you?
Well, GenSpace has a website, which is just genspace.org. And most of the links on the site link out. They link out to other resources, and they link out to, actually, I haven't done it yet, but it's going to link out to more of a personal page for me if people are interested in what I'm doing. We also are involved in this barcoding of Alaska project where I have a cabin that I built in Alaska. I fell in love with this remote area a few years ago, and a crazy friend of mine, Jamie Mansell, probably have heard of Mr. Giant Robot.
Yeah.
He's a friend of a neighbor of mine up there, and she convinced me to hire him to help me build this cabin, and we became very good friends.
We might have to get you back on.
This. Yeah, we built this cabin in 8 days. It's actually, he's got it on YouTube under Alaska Cabin, which is pretty funny. Of course, I did most of the filming, so it looks like I wasn't working, but I was working too. But it was pretty amazing. We actually felled the trees and stripped the bark and built a real log cabin in 8 days, which is pretty amazing.
Did he make you wear chains on your ankles while you were working or on your wrists?
No, he hadn't done that. That the chainmail stuff came later. Actually, I tried my hand at it, but I was kind of lousy at making chainmail. But so I have a geodesic sphere on his property in Vermont that is sort of half finished. That was my contribution to his group of his band of what does he call it, the Builders Club or something. I can't
remember, but yeah, something like that.
Right, right. The Adventure Builders Club. That's it. So.
It's amazing how many friends of Jamie's we actually know, and we still haven't met Jamie.
Yeah,
there are friends and there are friends. I mean, how many people have actually slept with the giant robot?
True. One of our other friends, Joe Grant, who I think I might have mentioned to you earlier. I think they did track in high school. Together.
They were on the running team together.
Brown, when Jamie was at Brown, yeah, he ran track for a while, and then he didn't like the way they were running it. He just sort of decided to stop. He's pretty amazing.
Yeah.
But I had for one winter, I had what I called my bum house inside Banana Building, which was I had this little house made out of pieces of insulation that I had like a tent pitched in. And so when it was like 15 degrees outside, it was about 45 in the tent, so I could sleep over in the robot room.
That's awesome.
Sounds like basically we could have brought up pretty much any topic with you and had an excellent hour show. So
Yeah.
thank you for joining us. We very much appreciate it.
Yeah.
We are out of time for this week.
Sad.
You guys can find this show every week, Thursday evening, ZombieTech.tv. There's an RSS feed if you want to automatically have it pushed down to your phone or iTunes or whatever, or you can just push play and listen to it on the page. It's super easy. That's it for us. We'll see you guys next week.
Bye.
Bye.
Bye.